Good evening, all. Is it audible?
Yes, sir.
Right. So yesterday discussed about endosperm formation and endosperm types and endosperm hostoria. And also apart from that, yesterday we discussed in part of double fertilization, embryo formation also, zygote, how to develop into embryo. Today we will discuss about embryo development in dicot and monocot. Dicot embryo development and monocot embryo development. Before that, dicot and monocot, let's see some of the differences between dicot and monocot embryos. So first of all, here the one side column given, are you able to see the PPT slide? Hello?
Yes, sir.
Right. So let me take pen. Right. So in first one, the character is number of coleoptile, position of plumule, position of cartiladons, coleoptile, coleorhiza, scutellum, and suspensor. These are the major characters. And apart from that, number of cartiladons also we can add another one. In dicot embryo, the number of cartiladons 2. Here there are two cartiladons. Two cartiladons. Actually, this is here. This is number of cartiladons. Number of cartiladons. So in dicot embryo, di means 2. So number of cartiladons 2. These two cartiladons attached to the embryonal axis we will discuss later. I will explain clearly after development of embryo in dicot. In mono means only one. Mono means only one cartiladan attached to the embryonal axis. Position of plumule. Plumule nothing but the primary shoot apex. That is called plumule. The plumule occurs distally, the apical part. But the plumule in monocot occurs lateral side. Position of cartiladons. Two cartiladons formed laterally. We will see diagram later. In diagram clearly we will see about where is plumule, number of cartiladons, position of cartiladons in dicot lateral, and in monocot only single single cartiladan occupies the terminal position. Position, cartiladons, dicot lateral side, monocot terminal side. Coleoptile and coleobryza. Coleoptile, it is the protection part of the plumule. Coleorhiza, protection part of the radicle. These both no protection in dicot embryo about plumule and around the radicle also no protection coat. So that's why in dicot embryo coleoptile absent and coleorhiza absent. But in monocot the envelope plumule is called by coleoptile. Coleoptile plumule, already told you the plumule covered with coleoptile and radicle protected by coleorhiza. The scutellum offset in dicot, actually the single cartiladan considered as a scutellum in monocots. Suspensor in dicot embryo, suspensor in both dicot and monocot suspensor present, but in dicot embryo large suspensor present. In monocot embryo small when compared with the dicot suspensor, monocot suspensor small in structure. So this is the small introduction about dicot and monocot embryo. Right? Now, so now let's discuss about the embryonic development. If you see embryo development, first of all, what is the initial cell to development of embryo? What is initial cell? Hello?
Yes, sir.
What is the initial cell?
Zygote.
Right, zygote. This is the zygote, which is diploid in state, right? Okay?
Okay, sir.
Right. In angiosperm, the zygote divides transversely and forms an apical cell or CA. Apical cell or CA and basal cell CB. Basal cell CB. So here the always the apical cell toward the chalaza side and basal cell CB toward the micropyle side. Don't get confused. CA, for example, this is rough diagram ovule. This is micropylar and this is chalaza. Already we discussed earlier classes. This is upper side micropylar and opposite chalaza side. So, for example, let me change color. For example, this is the zygote. This is the zygote form. After single cell, unicell zygote, after transfer section, two cells are formed. This is chalaza end. For example, this chalaza side that is called as CA, apical side, remember it. And about the micropylar side, this side, micropylar side that is called CB, basal cell. Okay? If micropylar upside, sometimes what happen, do you know anatropous ovule? And what happen in this? So this is micropylar. This is chalaza. After transfer section, now chalaza side called as CA, micropylar side called as CB, like this. So don't get confused about the C and CB. So anyway, in angiosperm, the zygote divides transversely and form an apical cell. Apical cell always CA, but the apical cell toward the chalaza side, remember it. And basal cell that is called CB. The CB toward the micropyle side. And the basal cell CB undergoes transfer division and gives rise to daughter cell. CB, the basal cell CB, for example, this is considered as a CB. The CB cell undergoes transfer division and gives rise to two daughter cell M and Si. Actually, not given here. If you take another diagram here, CA, basal cell CB undergoes transfer division, gives rise to two daughter cells. M and Ci, M cell and Ci cell. The apical cell divides longitudinally to transverse, or maybe the apical cell. This apical cell may be divide longitudinal division or transverse division, and a linear shape T-shaped pro-embryo formed. On this basis of mode of cell division in apical cell, the role of CA and CB, the scientist Maheshwari classified the mode of embryogenesis into two main types. That is the mainly the based on plane of division of mainly especially apical cells, the plane of division. Maheshwari classified this embryogenesis into five main types. They are a crucifer type, monograde type, brassicaceae type, annonaceae type, and ranunculaceae type, so on. So let's see in this type mainly the first of all, apical cell CA divide longitudinally in the two-celled embryo. The basal cell CB may not play an important role in embryogenesis. For example, here the different type of embryogenesis are there. In our book also given. In this, the most important monograded type, we will discuss later how the monograded type embryogenesis occur. Before that, you should remain only one thing, that is here where type. So remember the apical cell CA divide longitudinally in the two-celled embryo. Basal cell CB may not play an important role in embryogenesis. It may be completely absent in cruciferous type. Already we discussed in cruciferous type, monograded type, a different type of embryogenesis. If the basal cell CB not important in some type of embryogenesis, and in some type of embryogenesis only apical cell CA actively involved to formation of embryo in embryogenesis. In some type both apical CA cell and CB cell participate. CA and CB participate in the development of embryo. That is called osteroid type. So here the first only CA type involved, only CB type actively involved in cruciferous type. Sorry, basal cell may not play important role in embryogenesis or may be absent in cruciferous type. Cruciferous, you can see in book also, cruciferous type or monograded type. Both apical and basal cell actively involved to development of embryo. That is called asteroid type. That is called asteroid type. So anyway, here we will see the different stages in development of embryo. Capsula bursa. Capsula bursa in our book also, in our theory book also given capsula bursa embryo development. Let's see capsula bursa embryo development. In this diagram, what you see in this slide, that is capsula bursa development. Let's see in this the first apical cell CA. Apical cell CA divide transversely in the two-celled pro-embryo form. The basal cell CB plays a minor role, already told you earlier, minor role in later development of embryo. Here the basal cell, CB develops into suspensor. Actually here, actually this is micropilar side, CB, Chalaja side, this one, CA. In this type, what happened? Apical cells, sorry, basal cell develop into suspensor. CB, this is basal cell. This is divided transversely and finally formation as a suspensor. We can see here suspensor. And in suspensor, the last cells act as a astorial cell. Astorial cell, I will tell you what the function of astorial and suspensor. One minute. Sorry for inconvenience. Are you able to see the PPT slide?
Yes, sir.
Right. In our book also given the cruciferous type and monogrody. Uh, cruciferous type, monogrody type, or cruciferous monogrody type. Best example, Kapsula bursa. You see, Kapsula bursa is the zygote divide transversely. Zygote. This is zygote. Let me tell you. This is zygote. In examination also, you just write in development of dicot embryo only cruciferous type or monogoid type only. Do not write all type of development. Zygote divide transversely to form two-celled stage. They like this. The first cell, which is faces chalaza side, is known as apical cell (CA), and the second cell faces toward the micropyle side, that is called CB, CA, and CB, CA and CB, which is called proembryo. A proembryo is formed by longitudinal division in the apical cell and transfer division in the basal cell. So after what happened? After two-cell formation, CA, CB, right. After formation CA-CB two-celled stage, longitudinal division in the apical cell CA, longitudinal division apical cell CA, and transfer division in the basal cell, transfer division in the basal cell like this. As a result, another longitudinal division in the CA. Next, another longitudinal division takes place inside the CA. Here, another longitudinal division. As a result, the 1, 2, 3, 4 quadrant stages formed. So this is called tetrad. After formation tetrad, the transfer division takes place. Just simple. Don't get confused here. For example, CB, CU. What happened in apical cell? In apical cell, longitudinal division takes place. Okay, one longitudinal division. Transfer division in basal cell. Another longitudinal division in the CA. Another longitudinal in each cell, like this. Now, how many cells are formed here? In apical cell, CA, how many cells? 1, 2, 3, 4 cells are formed. This is called quadrant stage. So this is tetrad. Now this tetrad transformably divide and form octant. Transfer division means like this. Now count it above 4 and below 4. 5, 6, 7, 8. Quadrant form. Sorry, octant. Octant is formed after transfer division. The transfer division takes place in CB cell. And what happened in CB? Another transfer division takes place in CB cell. This transfer division continuation. One transfer division, next another transfer division, next another transfer division, another transfer division, like this. 6 to 10 cells are formed. 6 to 10 cells are formed in CB cells. The finally it developed as a suspensor. After 5 to 6 development, it is developed as suspensor cell. Suspensor cell, 6 to 10 cell suspensor takes place. Of all these cells, the topmost swells becomes as a larger. It is act as a hostorium like this. So these are the octant cells here, octant cell, and later the ear formation suspensor from CB cell, and apical most topmost cell act as a hostorial cell. Hostorial cell. Right. Later, the lower cells is known as hypophysis. Lower cells means this one, this cells, this type. That is act as a hypophysis. Hypophysis. Root cap and epidermis and cortex of root are formed from the hypophysis. So from this hypophysis region, what happened? The root cap formed. Remember it, root cap, epidermis, and cortex of root formed from the hypophysis. In the actin cells, actually this actin cell two types, we can see clearly two types. In two tiers of four cells of each, the upper tier, this one, from upper tier further developed into cotyledon and stem apex. Cotyledon, cotyledon, this upper part, cotyledon and stem apex form. Like this, finally, the complete embryo developed. So if you take the embryonic dicot embryo, here plumule, two cotyledons lateral side discussed earlier. These are the cotyledons. These are arranged to lateral side. Arranged to lateral side. And this is the suspensor. Already we discussed. This is radical. So what happened? The radical region developed from the lower tier of the CA cells. Out of 8 from lower side, octant. And actually, it is not to stop at octant. This octant also further divided and redivide like this. Divide and redivide and formation as a procambian region, ground tissue, dermatogens, they are formed finally. But before they are formed, from lower side, root development are developed. Root development, all root parts, root cap, epidermis, cortex, all and other root parts are developed from lower side. And above side plumule, what we just but here the plumule and cotyledons are developed. So here the radical portion developed and cotyledon developed, and here the plumule also developed like this, and from the CB cell, basal cell, suspensor, osteoid developed. Now this complete developed into embryo. Now this is the complete development of embryo, right? So finally, which are developed, which are differentiated into uh, some of the reason they are outer peribellum and an inner plerome. So which is developed by the further divisions. Due to some more division, peribellum and plerome, from the here remember from the peribellum and plerome, these are the completely the internal parts are formed. The internal parts may be cortex and steel. Both the cortex from peribellum, plerome, steel. Plerome forms the steel. Peribellum forms the cortex. As a result of divisions in the group of the cells, apical region, the embryo appears in the form of odd shape. It looks like some odd shape. Due to lack of sufficient space in the ovule, the embryo converts into horseshoe shape. When due to the seed coat pressure, it is developed, it is appear as a horseshoe shape. Thus, the mature embryo is formed with central stem apex and two cotyledon on either side. The type is called honagrade. as it occurs in family Onageraceae, dicot. This is given in our book, and this much you have to write in examination also. Is it clear? Hello?
Sir, once explain, sir.
Simple. Simple. First of all, zygote clear single cell. Hello?
Yes. Yes, sir.
It is divided into two cells, CB and CA. CB cell and CA cell. Okay?
First, upper part CB, sir. Basal cell.
Basal cell CB, apical cell CA. You don't get confused here. Maybe the CB and CA cell you can write like this also, like this. CA and CB. And you can write CB and CA. That is based on micropilar and chalaja. Chalaja side CB. Sorry. Chalaja side CA. Micropilar side CB. Hello?
Yes, sir.
Okay, simple thing. CA apical, right?
Mm.
CB basal. Enough?
Okay, so CB, the basal one.
Basal.
Mm.
CA apical one.
Okay, sir.
Apical one toward the chalaja side. Apical one toward the chalaja side.
Okay, sir.
Basal one toward the micropilar end. That is
Mm.
based on type of working. Okay? That's clear.
Okay, sir.
Right. Anyway, I have taken this one CB basal cell and this is CA. Two celled. What happened after formation two cells? Right? Two cell. In two cells, already this is CA apical. This is CB, right? Are you follow me?
Yes, sir.
Right. In CA, what happened? One vertical division takes place. The next division, CB, transfer division takes place. That is first step. Next step, what happened here?
First CA, sir.
Yes.
Okay, sir.
In next diagram, already transfer division, already here vertical division. Later, actually, how many cells present? Two cells. Here, two cells from
Yes.
CA. In CA, two cells and CB also two cells, right?
Yes, sir.
Right. Later, what happened? In CA, two cells, each cell undergoes to another vertical division. Another vertical division. Now, how many cells are formed? In CA,
Four. Four.
four. In CA, four cells are formed. This is called quadrant. No further divisions in CB. Next.
In CA, lateral also vertical division, sir. Longitudinal division.
No, transfer division. Let me draw. Let me draw. CB already in CA one vertical division follows another vertical division. Next transfer division takes place like this. Transfer division.
Okay.
This is completely CA, right? Now how many cells are formed in CA?
8.
8 cells are octant stage form. After formation octant, now CB cell continuously follows the transfer division. Only transfer. Transfer division. Another transfer division, another transfer division, another transfer division. Like this, number of cells, 5 to 6 cells are formed until formation 5 to 10 cells, 6 to 10 cells. Now it is completely developed into suspensor. The topmost developed as a large cell, it is developed as a half story, like this. Okay. Hello.
Yes, sir. Sir, once
Right.
again, sir.
Which one do you want? CB cell, CA development?
CB.
CB simple. CB actually CB always continuation transfer division. Remember it.
Okay, sir.
Transfer division first two, one to two cells are formed. Again transfer division, transfer. How many cells here? One, two, three, four. Again transfer division. That means four formation as a eight. Again transfer division 16 like this. So anyway, after formation, the number of cells either only by transfer division, only transfer division. While the division takes place, it is the grows upside direction. Hello.
Yes, sir.
Right after formation, only one direction, only transfer direction only. The
Okay.
finally almost 8 to 10 or 6 to 10 cells, that is the base of the species, 6 to 10 cells form. The all together called as a suspensor cells. In topmost cells in suspensor somewhat swollen, that is called Astorius cell. That's it. Next, what happened in CA cell? Actually, CA actant, right? What happened is further divisions also takes place. After formation, further division from the this, for example, this is the base A tier, upper B tier. From the A tier, remember root pod develop. In embryo, root pod develop. From B shoot pod develop. Simple. What happened here? Actually, here periclinal division takes place. Due to the periclinal division, here three tiers of cells form. Upper, central, cerebellum, peripellum. This is pleurum, inner pleurum. Simple. Let me. Should I draw again? Hello.
Yes, sir.
Just wait. Until formation, acton stays clear. That means root formation, shoot formation clear.
Yes, sir.
Right.
In CB, the
Yes. Yes.
lower part develops as root part,
Right.
and the upper part develops as shoot part.
Right. That's enough. That's enough. Just
And
wait.
this periclinal division, the
Right.
three are formed. What are they, sir?
Epidermis, epidermal cell, upper one, and pleurum, peripellum. Just wait. Just wait. I'm drawing only CB, sorry, CSL only. Don't get confused. Only CB. Sorry,
Okay, sir.
CA. Only CA.
Okay.
Formation octant. Here the periclinal division means this is periclinal division. Is it clear? How many tiles are formed due to periclinal division?
Three types.
One, two, three. So upper tier, this is forms the epidermis of shoot and root. Middle formation cortex, shoot and root, and inner formation steel. That's it. This is epidermal region. Cortex formation region is the peripellum. This is region peripellum. Peripellum in embryo. Embryonic, it is called as peripellum. This peripellum later forms as a cortex, while developed later formed as the cortex. Next, pleurum, inner one pleurum. Pleurum. This pleurum further developed into steel. Now, is it clear for periclinal division? Clear? Hello?
Yes, sir. Clear.
Right. Just wait. Let me take. Yellow color, blue color, green color. Right? Three regions are formed. Epi, the center one, periphelum, periphelum, which periphelum forms the cortex, center, inner one, pleuro. Pleuro forms the steam. When the embryo matured and formation as a plant, these internal parts are developed from the embryo. Actually, in embryonic, that is plerome, periphelum. The parts called as periphelum, not cortex. Plerome, not steel. But later, when it is developed, that is called periphelum. Periphelum is a cortex, plerome is a steel. That's it. So anyway, while it is developed into seed, what happened? That due to the pressure of the seed coat, it shows the curvature. It shows curvature nature, and it is appear as a horseshoe shape. That is the characteristic feature of monochromatic type, cruciferous type. So finally, it is complete developed into dicot embryo. We can see in dicot embryo development here. This is the root covered by root cap, radicle covered by root cap. This is hypocartil place and above epicartil and plumule shoot apex and root cartilage. So this is dicot embryo. Is it clear?
Yes, sir. Sir, here one doubt, sir.
Ah,
Sir, here upper side, how the hostorial cells formed, sir?
oh, 1 minute, 1 minute. Upper side,
In suspension cells,
okay, in suspensor cell. Ah, how the how
hostorial.
the how the osteorial cell formed? Just a minute. Ashwin, sorry for disturbance. Undoubtedly. How the osteorial?
Hostorial cells.
Your doubt is how osteorial cell formed, right?
Yes, sir.
Simple. What happened actually? Ostoria is the part of suspensor, right?
Yes, sir.
The suspensor formation by the repeated transfer division in CB cell. What happened actually initially, this is the suspensor cell. There is no ostoria.
Mm.
And here also in rest of the parts also developed, plerome, periphelum, all this developed. For taking nutrition, what happened? From taking nutrition from the, what is surrounding part? This is embryo, okay. This is embryo. What is surrounding part to the embryo?
Endosperm.
Right endosperm. To penetrate into endosperm, automatically the topmost cell, this one, automatically developed into enlarged, enlarged, and penetrate into endosperm and
Oh,
receive the food material from the endosperm and gives it to the developing embryo. That's it.
okay, sir.
That's how it is formed. Okay,
Okay, clear, sir.
right. So this is about dicot embryo development and dicot embryo structure. In examination, if they ask about dicot embryo, just write about cruciferous type. That's enough. Okay. Next, let's see the embryo development monocotyledon.
Okay,
Dicot
sir.
clear?
Yes, sir. Clear, sir.
Right. Now let's see embryo development in monocotyledon. Right. So this is structure of monocotyledon. Let's see. Embryogenesis in monocots is similar to dicot. Don't get confused here. The one word enough. Embryogenesis in monocots is similar to dicots up to formation of octant. Do you know how the octant formation? Hello?
Sir, yes, sir.
Right. Octant formation clear in dicot embryo development?
Yes, sir.
Right. So in monocot also, the similar way development takes place. In monocots, only single cotyledon occupy the stem apex. Only single cotyledon, as it monocots, only single cotyledon occupy the stem apex. According to Ward law, the plumule, which is lies at the apex of embryo, but in dicot lies lateral. According to Ward law, the plumule, which is lies at the apex of embryo of dicots, lies laterally in the embryo of monocots. Swami and his co-workers studied the embryogenesis in monocotyledon in detail. Let's see the development of monocotyledon in Sagittaria. In Sagittaria, different stages are formed. In Sagittaria, first of all, any type of embryogenesis, whether it is mono or di, first initial cell is zygote, right? Where it is right. Initial cell is zygote, which is zygote. Let's take color. Initial cell zygote. As a result of transfer division in zygote, the apical cell and basal cell formed, right? The same apical cell CA toward the chalaza side and CB toward the micropilar side. CA, CB both are formed, CA and CB. The basal cell expand extensively into sac-like structure without any further division. Sorry, which one? Basal cell, right? Here the basal cell expand extensively like a sac-like. So we can see in diagram also. Actually, what happened in dicot embryo? Dicot embryo CB, which division follows? Hello, Suhana.
Yes, sir.
In dicot embryogenesis, CB cell, what type of division takes place? Longitudinal
Trans
division or transfer division, right?
transfers.
Transfer division takes place. But in monocot, remember, no further divisions. Just the basal cell expand extensively. Extensively, like this. The basal cell expand like this. As a result, right, the sac-like structure formed. So due to the one longitudinal and transfer division in the terminal cell of the pro-embryo, quadrant formed. The later what happened? So it is finally settled as a suspensor. Okay, that is there's only single cell expanded. That is finally settled as suspensor, right? So in first in CSL like a first of all this is CB CA. This is expand as a sac-like structure and suspensor. And in CSL, one transfer division takes place. Now it is only three cell, one, two, three cell form. Later, due to longitudinal and transfer division in CSL, pro-embryonic quadrant and form. Longitudinal division, one, two, three, four cells are formed. One more transfer division takes place in tetrad. As a result, same, actually, you don't get confused here. Forget all this. Just wait. You remember one word, that is in CB, no further transfer division, directly they act as a suspensor. And remaining division, transfer division takes place in CSL quadrant form like a dicot, and octant also form. So after formation octant of these 8 cells, 2 tiles, 4 cells, right? Only CSL, I draw only CSL. Is this octant or not? Hello?
Yes, sir.
How many tires? Tire, this is one. A tire 1, B tire 1, 2 tires. Each tire 4 cells, right?
Yes, sir.
Uh, Suspense, okay. Same here also, periclinic division take place in octant. Outer, actually I called as a epidermis. The epidermal cells called as dermatogen. Dermatogen. Here also same 3 regions. Red, green. So this one, uppermost, this is called dermatogen. Actually, this one forget in dicot embryo. Dicot embryo also same. Upper, dermatogen. Periplum, middle, periplum. Center, what we called?
Pleroma.
Plerome. Right. Plerome. As a result of division, the inner cells, periplum and plerome cells are formed. This entire part develops and forms into single cartilagen. At the same, in dicotyledon, 2 cartilagens are formed. But here only single cartilagen formed. The stem apex is formed laterally in a constriction like an area between embryonal axis and cartilagen. Here, just wait. Wait, it is okay, right. This is no need. This one, this is cartilagen. Later, after well developed the embryo, this is cartilagen, scutellum part. This is cartilagen. This is plumule. Remember here, the only one different, the entire part develop and forms into single cartilagen. This entire part developed into single cartilagen. The stem apex is formed laterally. Apex in a constriction like area between embryonal axis. This is embryonal axis and cartilagen. This is cartilagen. So between embryonal axis and cartilagen, small constriction formed. This is small constriction where the plumule develop. So that's why the plumule, the apical region, and the plumule somewhat slightly lateral side, but in apical side, here the only apical side, one cartilagen present. The plumule or shoot apex somewhat slightly lateral side. For example, which is axis in clear view. This is single cartilagen where here the plumule, shoot apex, plumule present. And here common, this is radical. The radical covered by chorioryza and shoot apex, this one covered by coleoptile. This is by coleoptile radical. Actually, this coleorhiza, coleoptile not formed in dicot embryo. The same development, you just write the same development where we see the dicot embryogenesis, but end in monocotyledon, just small changes. The small changes are here the number of formation cotyledons only one. And after entire development, the radical covered by coleorhiza and plumule covered by coleoptiles. Coleoptile, that's it. Is it clear?
Yes, sir.
Yes, you can write the same development. Don't get confused here. Same development as you learn in dicot embryogenesis, you can write same. But only
Sir,
one
here,
difference. Ah,
sir, here Hostoria cells.
here Astoria cells, Astoria maybe embryo sagittaria, where the Astoria cells, Astoria suspensor, okay. Of course, Astoria also should be formed here, but not given.
Okay, sir.
Actually, let me check 1 minute. Astoria formation. What is function of Astoria?
It, it provides the nutrients to the embryo.
Right, right. Monocot embryo. Right. Actually, actually, no further transfer division. Only one cell enlarged as a sac. That is called suspensor. Astoria monocot. Where it is? The cells produced by the basal cell, which located toward the micropore. Okay, make it as serve as a astoria. It has the and toward the micropolar and big, thick and swollen. Actually, here the remember the entire suspensor cell act as a astoria.
Okay,
That
sir.
is suspensor and it is act as a astoria. But what happened in dicot? In dicot, not only single cell, more than 6 cells are there. But
Yes,
all
sir.
are suspensor except the topmost. Of course, the topmost cell also suspensor cell, but somewhat it's swollen. It is considered as a hostoria. That's it.
Yes, sir.
Only this in dicot embryo, only the hostoria penetrate into the endosperm. But
Endosperm.
but in monocot, the entire enlarged cell penetrate into the endosperm. That's it.
Endosperm. Okay,
Right?
sir. Yes,
So
sir.
now next one, another important. These two important, dicot embryo development, monocot embryo entry. Compulsory either dicot embryo or monocot embryo. Dicot embryo or monocot embryo. And another
Okay, sir.
important one is polyembryony. Polyembryony. I don't want to waste your time and my time. Just simply you tell me what is polyembryony. Already you see dicot and monocot embryo. Now tell me in general definition what is polyembryony. What is meaning of poly?
Many.
Many. So in polyembryony, formation of embryo more than two, right?
Yes, sir.
Right. Just wait. Already we know how the embryo developed. Just wait. Let me take the slide, a whiteboard. Right. So just you remember the embryo formation by very sketch, color, right? Right. So embryo formation developed by, hello,
Sir, yes, sir.
how embryo formed. For example, in ovule, this is embryo sac, this is the female gamete, and these are the synergids, secondary nuclei, antipodals, right? So this is female gamete, plus one, female gamete. Now, if the female gamete, when the fuse with male gamete, that is developed zygote embryo, right? But how many embryo formed here?
One.
One, that is true. But in polyembryony, more than one embryos are formed. Actually, this is the character feature of gymnosperms. What happened in gymnosperm, and also we can observe in some angiosperms also. This polyembryony, mainly two types. One is true polyembryony. Another one is false polyembryony. False polyembryony. First, let's see the true polyembryony. In true polyembryony, embryos are formed due to the cleavage of zygote. Of course, the of formation of embryo common, like inside the endosperm. Sorry, inside the female gametophyte or embryo sac. In especially In female gamete, after fusion of this is male gamete fused with female gamete, as a result formation of zygote, diploid zygote. Right? How many cells? Unicell. After this diploid zygote further developed into embryo. Embryo means number of cells already we discussed. Discuss number of cells are formed. After formation, this embryo, which is further developed into root system and shoot system. That's enough. But what happened in true polyembryony? After embryo formation, cleavage takes place. Cleavage means after formation, this group of cells, this is separated as a one group of embryo. This is developed into one group of embryo. This is developed into one group. How many embryos are formed now? Blue color spots. How many?
3.
Three. Now this is developed into embryo. This is developed into. This is developed into embryo. So there's a true polyembryo. And next, what is false polyembryo? False polyembryo means if the embryos are formed from two or more embryos